I'd like to welcome all of you here today, and everybody in this audience has actually played a pivotal part in this project. And it's interesting because I am now part of this group that's kind of over the hill. And so for me, it was a real pleasure to actually be involved in the research side of things because at my age, it's past the 60 mark, it's actually a particularly valuable exercise too. So science is my bag. My background is that I grew up on a farm in rural Manitoba in a place called Pipestone, which most even Manitobans haven't heard of because it's got about 150, 60 people in it. And so it's a real proud moment to be able to, I wish my mom and dad were alive because I think they would be very happy to see that the Made in Manitoba project has their Made in Manitoba daughter participating in it. And the other thing, science is really important to me, and I've been involved in science since I graduated with my Ph.D. for over 30-some years, but at the same time, I think we all have to have a sense of humor. So I hope you do too, because I'm going to share a little anecdote with you about myself, which is one of the more dubious distinctions I've earned over the years. A number of years ago, my background is that I'm a clinical microbiologist, and my research interest has been hospital-acquired infections and how to prevent them, and also the microbiome and trying to find ways to improve it. And Clostridium difficile, or C. difficile, is one of the diseases that can be caused in the gut, and it's a real problem, especially in the elderly population. So I've been doing research in this area for a number of years, and at one of the microbiology conferences a few years ago, I got called up to the stage, and I didn't know why they were calling me up to the stage, but they said they had this wonderful award to present to me. And I'm thinking, oh, you know, what's going on? Because these are my friends calling me up to the stage in front of this audience of several hundred microbiologists from across Canada. And I got up there, and my colleagues presented me with this wonderful tiara crown and a toilet plunger, and I was awarded with the Queen of Poop Award. Because most of my studies had involved doing fecal samples, and so to the participants, you can relate to this. So you didn't know that you were somehow related to the Queen of Poop, but indeed you are. And on that note, I would like to actually go through some of the results from this project, and hopefully you'll be as excited about the results, because I know the participants wouldn't know the results at this point. So we've kind of talked about this, the Made in Manitoba project. I'm not going to go over this anymore. Pram and Earl have done a wonderful job actually acknowledging all the individuals that were participating in this project. And it really does take a village to actually do a clinical study. So myself, I would like to certainly acknowledge Pat DeGagne and Nancy Olson, my tried, trusted two right-hand people that are in the audience and have been with me ever since I started working at St. Boniface Research Center. And I really do appreciate and acknowledge the big role they played in my research career. The Deer Lodge participants, Dr. Strang was the clinical investigator, the physician for our research study, and I wanted to clarify that some of our elderly population, and they're the 70 years plus individuals, were residents at Deer Lodge and would really like to acknowledge his role in our project, Dr. Strang's, but also the Deer Lodge staff that were unbelievably supportive. Now, we couldn't fulfill all of our enrollment at Deer Lodge, and so many of our elderly participants were community dwelling individuals that participated in the project as well. And then, of course, our 30 to 50-year-old participants were recruited from the community base as well. St. Boniface Hospital, you've heard about the huge involvement of many different individuals in that. The National Microbiology Lab, suffice it to say that without the support from the National Microbiology and the University of Manitoba Statistical Analysis Unit, some of this data would still be being analyzed at this point. So many thanks to all of the participants, but also to all of the people in the back that played a role in this study. So let's have a look at this. I don't know about you, but sometimes it's extremely confusing understanding what gut health really is and what it's all about. So I found this fascinating many, many years ago when I identified this. There are more microbes, more bacteria in our gut, in our feces and our bowel than there are human cells in our body. And if you think about that for a minute, it doesn't take rocket science to understand that those many microbes in our GI system probably play a really important role in many aspects of our health and well-being. Just ask anybody whose bowel is not working properly the impact it has on you and how miserable your life can be when things aren't going well. So there are more microbes in our body than human cells, and they do play a very important role in our health and well-being. Now, one of the interesting things, when I talk about microbiome, it's a big science word, but basically what we're saying, it's a fancy way of saying that the community of bacteria, fungi, viruses, etc. that are there make up a microbiome. So it's like saying the world is a microbiome of people. Well, in our gut, we have a gut microbiome. And a lot of people don't quite realize that 10 to 15% of the energy we require each day to be active, that energy is not made by our body. It comes from the end products of the bacteria in our gut. And so it is an important source of energy for our bodies, the type of microbiome that we have. The microbiome in our gut also provides critical nutrients that we cannot provide for ourselves. So for example, vitamin B12 is a crucially required component in the human body for it to survive, and that component is actually not made by us, it's made by the bacteria in our gut. And if we didn't have them, we would actually not survive because we need vitamin B12. The other thing that it can do, it digests some components of food that our body digestive system cannot properly degrade. So for instance, if you take a plant, a vegetable, a fruit, a raw one, many of the fibrous components in there, our body can't degrade and utilize and convert to energy. But the bacteria in our gut can digest them and release the energy so that we can actually get a benefit from those. Now, the other thing that's interesting to me is that the microbiome changes, and I'm going to go over this in a little bit more detail in the next slide. We start off with nothing, we get a stable microbiome, but toward as we get elderly, there is a deterioration of the microbiome, and it's thought to be related to the diet, and in particular to the lack of digestion-resistant starch. And if you look at the next slide, it's really interesting. When we're born, we don't have any bacteria in our gastrointestinal tract, and over the next 10 years, we're actually developing our gut ecosystem. By the time we reach about 10 years old, we establish a stable gut bacterial mix, which is resilient, and you can see that it stays pretty stable for most of our life until we hit that magic 60 years of age mark, and then things tend to deteriorate. But I'm hoping I'm going to shed some light onto ways that we can try to change that a little bit at least. And so at the adult microbiome, Firmicutes, Bacteroidetes, Actinobacteria, Proteobacteria, I love saying their names, but I'm a microbiologist, and they're very annoying to people that are not microbiologists, and it's intimidating. So I'd like to make the analogy. These are the big groups of categories of organism that are present in our gut. And so if you think about it, it's like the analogy in the world. We have Canadians, we have Italians, we have Americans, and in a way I think of these as kind of those types of groupings. So when I talk about the Firmicutes, they represent 50 to 80% of all the bacteria in our gut microbiome. And that's followed by the Bacteroidetes. So if you think of it, we're Canadians, right? We're the Firmicutes, we're the good bacteria. So we're representing 50 to 80% of the gut bacteria. And I'm not saying this negatively, but the Americans come second. They have Bacteroidetes, and they represent about 10 to 15% of the microbes in our gut microbiome. And then we have the Actinobacteria. They're from far abroad. They come from South America, and there's fewer of them in our gut. And the Proteobacteria only represent about 1 to 20%. So I don't know, I guess they come from the North Pole or Antarctica, something like that. Think of it that way. And that population in our gut is pretty stable up until around 60. And then it just depends on the country you're in, the diet you have, and you as an individual, because we all change differently. Not the same things happen to everybody at the same rate. But about that time, there starts to be a decrease in the diversity of bacteria and microbes in our gut microbiome, and we get an aged type microbiota. The Firmicutes are decreased, and when that happens, it's those Firmicutes that can digest the digestion-resistant starches. And when we get a decrease in them, we have less of an ability to digest that type of intake that we have. And the other thing that happens is there's an impact on the intestinal motility. As we age, we have more problems. So constipation becomes a big topic of conversation in individuals that are 70 years and older. And the other thing that happens is there's an increase in the gut permeability. It becomes less of a good barrier, and some of the function that it plays deteriorate. So what we're looking at, we really wanted to evaluate this aspect of the microbiome and determine whether there's any way to modulate that in a way that would improve some of the health and the health benefits. So that's kind of talking about the gut microbiome. Now, I don't know about you, but I'm inundated, and sometimes when I read literature, it's hard to understand what these terms mean. We've all heard of probiotics, and we've also, you may have heard of prebiotics. And what's the difference between those two? If you think about it, probiotics, this is a term that's actually provided for some of the products that have live bacteria in them, and they're the good bacteria. And examples of that would be the bifidobacterium and the lactobacilli. So if you eat a yogurt, and I'm not supporting any of these companies or anything, I'm just using these as examples of the types of sources where you can get probiotics, but yogurts often have bacteria present in them, and there's a number of different nutritional supplements that you can buy as pills, and in actuality, when you eat any of these products, you're eating live bacteria. And the live bacteria are called probiotics because the ones in the mix that you're being given in these forms are things like lactobacilli, bifidobacteria. One thing that you may or may not be aware of is that they are transient. They do not permanently colonize your gastrointestinal tract. So the moment you stop consuming your yogurt or stop taking your probiotic pills, those organisms will disappear from your gastrointestinal tract, and it will revert to whatever your normal situation was for the microbiome. Now, the other interesting aspect are the prebiotics. What are they? They're actually complex carbohydrates. They're the plant, vegetable, and fruit components that we ourselves cannot digest, but the bacteria in our gut, some of them, not all of them, but some of them can digest it. And by digesting these components, basically you are stimulating the good bacteria in your gut. And it does provide health benefit. There's a number of studies that have actually shown that by stimulating good levels of bifidobacteria and lactobacilli, you do get a health benefit from it. So if you think about it simplistically, prebiotics are essentially food for the probiotics, and the probiotics are good bacteria that either exist already in our gastrointestinal tract, and remember, the ones that exist in your gastrointestinal tract, they're with you for life. And so if you can stimulate them, that's fantastic. If you can't, you can always eat components that have probiotics in them, like yogurt, which is a very good health supplement. And the prebiotics, or the components in the non-digestible starches that are present, feed these good bacteria that break them down. They subsequently ferment them to produce components like short-chain fatty acids that benefit our gut. Now, one thing you may not realize, and even me as a microbiologist until I started kind of doing research in this area, our gut, the colonocytes on the inside of our intestinal tract, their main source of energy, believe it or not, is from bacterial short-chain fatty acids. So without those short-chain fatty acids, our colonocytes actually deteriorate and can die. So you can see what I'm describing is a symbiotic relationship. The bacteria in our gut, the good ones, do good things for us. We benefit from them. And I guess what we're doing is providing them with food that they can eat and stay in the ecosystem. And in the end, we have a nice, happy, healthy gut. Not always that way, but that's kind of the role that prebiotics and probiotics play in terms of trying to keep gut health. So let's look at digestion-resistant starch. What does that actually mean? Now, the interesting thing is, if you look at the Health Canada food guide, you will see that the dietary fiber intake that's recommended is between 25 to 30 grams of dietary fiber per day. And if you look at the information that's been gathered on Canadians, on average, we consume between 3 to 8 grams of dietary resistant or digestion resistant starch per day. We're way under what we should be. Now, this particular product that we are studying in our clinical trial, MSPrebiotic, is a digestion-resistant starch, and 10 grams, which is about 2 tablespoons of this product, provides about 7 grams of functional fiber. So this product not only has health benefits that we're going to see, and I will explain them to you, but it also provides enough to meet some of the dietary fiber requirements. And those participants in the audience that are here today, you were consuming 30 grams a day of this particular product, and that was giving you 21 grams of dietary fiber. So you were getting pretty close to what is the recommended amount suggested for Canadians and adults. The one other thing that I wanted to mention is the digestion-resistant starch that we were working with is heat sensitive. When you heat the component, you convert it to a digestible form. So for example, if a vegetable like a potato or broccoli or any other vegetable is cooked, you lose a lot of the digestion-resistant starch, and it becomes a digestible form that doesn't give you the benefit of the raw components from those vegetables or fruits. So your mums were right. Eat more raw vegetables, more fruits, and things like that, because you are getting a benefit, and your bacteria are benefiting from it as well. This is important because what it meant was when we were doing the clinical trial, we couldn't heat the product that we were testing and put it in a food, and we couldn't apply it to hot food products. So you couldn't mix it in hot oatmeal, for example. It had to be taken either as a cool liquid or something that was room temperature. So that's why I'm mentioning it at this point. Now, the objective of our clinical study was to look at the impact on gut health when MSPrebiotic, which is a powder which had not been categorized or hadn't had any clinical studies in humans done on it, and we weren't sure whether it would meet the criteria of being a prebiotic, one of those words I used before, and that's part of what this study was about. Could it meet the definition? And the definition of a prebiotic is that it has to show a change to the microbes in your gut or wherever you're investigating, and it has to be associated with some health benefit. Those are kind of the criteria for calling something a prebiotic. So what we looked at, and we were targeting the elderly, because you notice in that chart, the elderly are the ones that have those microbiome changes, and we were trying to see if we could actually offset some of those negative changes that happen in the elderly population. And we wanted to look at bacterial changes in stool samples. We also wanted to, two other things that we looked at, and there were more, but I won't talk about them today, are the glucose and inflammatory markers in the blood. And we wanted to compare it to a group of adults between the ages of 30 to 50 years of age. And you might ask, okay, Michelle, why did you include that population? And we did because there's data in the published literature that suggests there are big differences between the gut microbiome and some parameters in the younger age group compared to the elder age group. But there's no data out there on Canadians doing that type of comparison. So we wanted to make sure we included both age groups in our study and determine, are there really some parameters that are quite different once you reach 70 years and older compared to a younger age group? Now, those who participated in the study know this. There was a lot of exclusion criteria, and because our study was aimed at looking at relatively healthy individuals and assessing what impact this digestion-resistant starch had. So any of these would mean you could not participate in the study. Any gut-related diseases, pre-diabetes, diabetes, pregnancy, probiotic supplements, et cetera, and particularly antibiotics, because we know antibiotics kill bacteria in the gut, and we didn't want that confusing the issue when we were looking at what the microbiome changes were. So with these exclusion criteria, there were a number more, I couldn't fit them all on the slide, the populations were recruited. And there were 4 arms in our study. 2 of these, arms 1 and 2, involved individuals that were 70 years and older, and there were arms 3 and 4, which involved the age groups 30 to 50 years old. And we had determined at the beginning that we needed a minimum of at least 20 individuals in each of those arms to be able to analyze the data in a statistically solid fashion. These numbers show you the number that did get enrolled in each of the arms, and this number are the number that fully completed the full 14 weeks of the clinical trial. So we enrolled a lot more people than this, but there were 22 enrolled that completed the MSPrebiotic for the 70-year-olds, 20 that completed the placebo arm, and the placebo arm is a very important arm. The placebo was amioca, it's a corn type of starch. but it was packaged and looks exactly the same as the digestion-resistant starch we were evaluating, and the packages used, the participants would not be able to tell which arm they had been randomized to because everything looked pretty much identical. The difference being the placebo is totally digestible, MSPrebiotic is a digestion-resistant starch. And so with these groups in mind, at the very beginning of the study, we took samples of stool and blood because we wanted to know, before we start taking any product, what's the baseline information on these individuals. And then after 2 weeks, for 2 weeks, everybody consumed placebo, and then at week 2, they were randomized to one or the other arm of the study. And you might ask, well, why did you have everybody take placebo for 2 weeks? We did this because if you think about it, we're not used to putting powder in our food products or taking it, and we wanted to make sure that all the participants were familiar with how to use the product, how to consume it, before we started randomizing them to the 2 arms of the study. And then after that, we took samples at 2 weeks, at 6 weeks, 10 weeks, and 14 weeks. We took blood samples and stool samples, basically 5 of each of those from each of the participants in this study. All right. Now, the details of this are that the placebo versus the MSPrebiotic, the digestion-resistant starch, 30 grams of powder was ingested daily, cannot be heated, as I mentioned previously. Each of the participants had to fill in a detailed gut health record, and in it we asked questions like, did you take antibiotics today? How many bowel movements? I'm sure, I don't know if you know or not, the participants know this, but there is a Bristol stool scoring system, and it shows you pictures of what poop looks like. And it's categorized on a scale of 1 to 10. Is it stiff and rock hard, or is it runny as can be? So the lovely participants in our study had to score each day what their stool looked like when they had a bowel movement. Very exciting being involved in research studies. And from these stool samples, we actually analyzed the microbiome to assess what type of microbes were present. And we also analyzed things like the short-chain fatty acids that I mentioned were important for the good health of the colonocytes in your bowel. And in the blood, we assessed pro-inflammatory markers, lipid profile, glucose, particle size, and insulin resistance. I'm not presenting all the data today because you know what they say, never get a professor up in front of you and start talking about research because they'll talk forever. So I'm limiting myself, and hopefully we'll be done in a good time. But this is kind of an overview of what we did with each of those samples that we collected. So the primary markers that I want to share with you today from the stool samples were the microbiome results. And I really want to acknowledge and thank the National Microbiology Lab. I'm not going to go into any of the details of how they did it. Suffice it to say, it's like a wonderful show on the CSI series where they extract the genetic material from the poop sample, and they basically work with sequencing methodologies, and what they can do is identify all the types of microbes present in that stool sample without having to do culture, because culture is very difficult and it's hard to quantitate. And what we wanted to really look at, we looked at all the bacteria that were present, every one of them. But the ones I'm going to focus on today are, we wanted to know how many of the good bacteria like bifidobacteria and lactobacilli were present. Did that change during the course of the placebo or the MSPrebiotic? And how many of the bad bacteria were present? And so the proteobacteria, that group from, I think that was, I can't remember, that was the one from South America, I think. They're okay as long as they're there in reasonable numbers, but if their numbers increase, they will stimulate inflammation in the gut. And so you want to make sure that the quote-unquote bad bacteria levels are not too high. And then the secondary markers I want to share with you are the inflammatory markers from the bloodstream and glucose metabolism, because we found some very interesting information from those types of markers. So now we get into the science. I know this looks intimidating. The graphs always, drive people insane. And if it was an engineering graph as a microbiologist, I'd be saying, what the heck is this all about? But be patient. We're going to walk our way through it. So if we look at the beginning, this was when they were enrolled before they took any of the product. And what we wanted to know was, were there differences in the microbiome between the elderly participants versus the mid-age participants? And again, here's our big groupings: Actinobacteria, Bacteroidetes, Firmicutes, Proteobacteria, and a few others. And what I'd like to draw your attention to is the Proteobacteria family. In the ELD, before we gave them any product, we found that there was a statistically significant increase in abundance of these proteobacteria compared to the same group of bacteria in the mid group. And you can see the arrows are pointing out, but it's statistically significant as well, that there was an elevated amount of these proteobacteria. And when we investigated and said, okay, we know the proteobacteria group is elevated, but what members within that group are the ones that are increased in number? And so we wanted to check in that grouping of bad bacteria, which were the ones that were causing the increase. And what we found, it's a dysbiosis in the elderly. And why is it called a dysbiosis? Because it's a shift in the abundance of organisms that are known to have inflammatory effect on the bowel wall. And what we found was that the members of the proteobacteria that were increased were the Escherichia shigella group. Now, these are showing the most abundant organisms that are present, not necessarily only from the proteobacteria group, but from all of the groupings. But this is the member of the proteobacteria group that is there in elevated levels. It's that group. And I'd like you to look at the lactobacilli group beside it. And remember I said lactobacilli are members of the good bacteria? Well, you can see the proteobacteria actually outnumber those lactobacilli at the beginning in the microbiome for the elderly population. The other good bacteria are the bifidobacteria, and you can see we have bifidobacteria in the elderly microbiome, but again, they are not some of the main players. They are kind of more at a lower level. So this gives us a sense that yes, it's true, and this had been described previously, that there is a dysbiosis in the microbiome in the elderly population. And if you remember that graph at the beginning where it talks about the deterioration, and one of the things that is associated with that has been reported, the increase in proteobacteria. So let's look at what happens when those elderly population are on MSPrebiotic, the digestion-resistant starch, or they're on placebo. And you can see the data here with the red line. The proteobacteria in the group at the end of the study actually has very low levels of the proteobacteria, and the group that were on the placebo still have a much higher level of proteobacteria. So the digestion-resistant starch was actually able to modulate the proteobacteria abundance and bring it down more in line with what would be present in a 30 to 50-year-old, which is good because it reduces the risk of inflammation in the bowel from these organisms. There's many other reasons for inflammation, but this is one that can cause inflammation. That was one of the findings in the study. And the other one that was actually, I have to tell you this story because at the beginning when we started this study, I'm a science person, right? And you got to show me the data before I'll believe that some of these nutritional supplements actually do anything for you. And so we started the study. I didn't know any of these results until the study was totally finished. So as there were some nights, I had sleepless nights freaking out thinking, oh my God, what if nothing turns out from this study and we don't see anything from it? It's going to be a huge amount of everybody's time, effort, and money, and we'll have nothing to show for the benefit. Well, I've shown you one of the benefits already. This one was really quite amazing as far as I'm concerned. And when we look at the elderly, those individuals on MSPrebiotic, we found that there was actually a significant increase in the levels of actinobacteria compared to those on the placebo group, and that this change was even more dramatic for the middle age group. Now, if you remember, that middle age group we included because we just wanted to make sure we had a good control group to compare the elderly to. We weren't necessarily expecting there would be a big change in their microbiome, but indeed, we actually found a very significant increase in the abundance of actinobacteria compared to those that were on placebo. And although I'm showing you the numbers and telling you it's significant, the reality is that you wouldn't know this until you did the statistical analysis, but this is a very significant increase in the abundance in both mid-age and elderly individuals in that grouping called actinobacteria. And if we look at the actinobacteria, the member in it, the good bacteria in it that was increased, was actually the bifidobacteria. So this is, and I mean, sorry I get excited about microbes and stuff, but this is the exciting part because what this means is, even when we don't give probiotic supplementation, we have bifidobacteria inside our gut that, albeit they're at low levels, we can stimulate them with feeding them the prebiotic. And to me that was very exciting because one of the things we didn't know was, do you need both a probiotic and a prebiotic to get the best effect? And this data would suggest that the prebiotic, this particular one anyway, can stimulate our endogenous bifidobacteria, and that to me was very exciting. And it does it in both the mid and the elderly age group, which again was very exciting. So forgive me, what kind of a microbiologist would I be if I didn't list a bunch of bacterial names? These are the bifidobacteria species that are present. And you can see that there's arrows here with 4 and arrows here at the side. These are the 4 species of bifidobacteria. The 4 lines here basically represent those species that actually increased within the bifidobacteria family. And one of the ones that's quite interesting also decreased. Bifidobacteria dentium, which is associated with dental caries, actually decreased from baseline. It's 25% of all the bifido species are dentium in the mid-age group, 46% in the elderly, and you can see that on the group that do MSPrebiotic, both the mid and the elderly, there was a big drop in this. And it suggests that the dentium ones associated with caries actually decrease as people are taking this product. Don't know the impacts of that, but it is an interesting observation. And it may be that the reason it's so high, 46% of all the bifido are dentium, it might be related to, as people get older, they have problems with their dentition, etc. So this data was actually published, and this is the publication that it came out in and the link that is there. So this is actually now a published article in the Clinical Nutrition Journal, which is one of the best types of journals that this data gets published in. And the last thing I want to touch on are the secondary markers from the blood samples, the inflammatory markers, and the glucose metabolism. And at the beginning, before taking any product, what we found was the C-reactive protein and the TNF-alpha. These are both non-specific markers of inflammation. And it's interesting that compared to the average or the mean in the mid-age population, the ELD have a much higher level of these non-specific inflammatory markers. And that suggests that there is some non-specific inflammation going on in them. And in fact, the TNF-alpha levels were statistically significantly higher in the ELD group compared to the mid group, and they actually are slightly above the cutoff for what would be considered to be normal or abnormal. Now you might ask, okay, so what does that mean in the overall scheme of things? There was no change in these inflammatory markers even at the end of the study. So the consumption of placebo or MSPrebiotic didn't change these parameters, but I wanted to point them out because it does suggest that what was published, that there is more inflammatory, non-specific reactions going on in the elderly body is actually true. And the basis of it, our study wasn't designed to figure out what the basis of it was, but it's something worth looking at in future studies. And why would we be worried about this anyway? We're worried about it because normally the healthy gut is a barrier, and it ensures that all of the bacteria and other components are kept inside the bowel. If you have chronic inflammation, and there's evidence to suggest that those proteobacteria provide chronic inflammation to the butt. Sorry, I'm losing it. To the gut bowel. And what happens is you get a breakdown of the barrier, and you get components going inside past the gut wall that normally wouldn't be allowed to do that. And the other thing that it does is it leads to glucose dysregulation. And so those components have been shown to set the scene for potentially going on to develop type 2 diabetes. And that leads me into the very last piece of information I want to share with you, and that was the fact that in the elderly, we found that there was a significant reduction in the glucose levels on people taking the MSPrebiotic compared to placebo. So the blue line represents those who are randomized to the MSPrebiotic, and you can see that over the 14 weeks there was a gradual decrease in the mean glucose levels, and this was significantly lower than what happened with the placebo group, which is shown in the red line. And the other thing that I haven't shown on this graph, in order to regulate glucose metabolism, insulin is very important in that role. And if you get insulin resistance, it means that the insulin cannot do its job as well. And what we found was not only did the glucose levels decrease over time with the MSPrebiotic, but the insulin resistance decreased too. both suggesters that this might help people keep their glucose metabolism at the right level. So the significance is, if you can't control these parameters, it can actually set the scene and lead to type 2 diabetes. And this phenomena, type 2 diabetes, is rampant in many populations around the world actually, but certainly in North America. And the MSPrebiotic might be able to reduce this, but obviously further clinical studies are actually needed in that population because that wasn't our population that we studied. So all this data has been submitted to a publication in the PLOS One journal, and we're hoping to hear back from them soon, and that it will come out as published information as well. So to summarize, the MSPrebiotic did have the effect of reducing the quote-unquote bad bacteria, the proteobacteria, in the ELD, not in the younger population, because they didn't have any increased amount of those organisms to begin with. It also reduced glucose and insulin resistance in the elderly population, which is a significant finding and needs more studies. But it also, MSPrebiotic, increased the bifidobacteria in the gut of both ELD and mid-age. And so what that did was provide us with the information to say that MSPrebiotic does meet the criteria of a prebiotic, and it does seem to be a valuable supplement for both ELD and mid-aged individuals. So on a note from ourselves in the CCARM group here in the Research Center, it's the first commercial product that's come out of one of the clinical trials in the CCARM group, and we're very proud and excited about that. And you can check the website I've indicated here if you want more information. So my take-home message is, remember, not that Michelle's the queen of poop, but that happy gut bacteria lead to happy, healthy people. And it is important that we look and make sure that our health of our gut is looked after, especially as we are aging. And I would like to take this opportunity personally. Everybody else has thanked the participants, but I would definitely like to thank them extremely as much as I can, because without those individuals who volunteered to be in this study, we wouldn't have been able to make these findings. And I did want to mention that the presentation, there are handouts that you can pick up on your way out, but also on our website, if you go there, you can actually see copies of these slides as well. So on that note, I'll stop and thank you very much.